Once they get in, though, in order to stay in, they've got to have some sort of establishment. How do they establish an infection? We know our body is very good at getting rid of things. How is it going to get in and stay in? We've talked a little bit about this already with viruses and those glycoprotein spikes. They have to attach. So let's talk about adhesion and attachment in the role of infection from that. All right. So Protozoans, helmets. So the helmets are our worms. Remember, protozoans, larger creatures that actually use some sort of structure like a disc. This is a protozoan. This is Giardia, actually, that causes the disease Giardia. And this is Giardia causes the disease giardiasis. And this has this structure that is an adhesive disc on it. It gets in, you find it in water. It's very common in North America. You drink contaminated water from a stream or a river, and you consume the protozoan cysts. And the protozoan cysts get through your gastrointestinal tract and develop in your intestines and become these protozoans, and they adhere to the mucous membranes in your intestines. That happens. Worms, when you talk about tapeworms, they have hooks or suckers that can attach from that. All right, so protozoans, worms, they use some sort of adhesive structure to get in and, or once they're in, to attach and stay in. What else? We've already talked about viruses. Viruses have those glycoprotein spikes. Bacteria also have proteins that allow it to adhere to cells in the cytoplasmic membrane. You'll hear to those referred to as ligands. Ligands are proteins on the surface of the microorganism that bind to proteins or glycoproteins on the surface of a host cell membrane. This allows it to get in and establish an infection, gets into the host cells that it needs to reproduce. All right, if just like viruses, the type of glycoprotein it has, the type of spikes it has, controls what type of host it can invade. So a bacteriophage are viruses that infect bacteria. Their spikes only bind to proteins that are found on a bacteria cell. Therefore, they can't invade humans directly. Same thing with bacteria. Depends on what type of ligands they have on their surface as to what type of host cell they need. So our SARS-CoV-2 virus, it has a particular protein, glycoprotein, a spike on its surface, and it binds to a particular protein that are found on the membranes of our respiratory systems, our mucous membranes and into our lungs. It's called an ACE2 receptor, and that is why it infects in the lungs. It's why the signs and symptoms of the disease involve respiratory problems. So that's what's going on. Now, if we lose those ligands on the surface, if there's mutations in those ligands on the bacteria or mutations in those glycoprotein spikes on the surface of the virus, then the microbe cannot invade the host. This is called avirulence. It cannot, we talked about virulence factors, things that allow it to attach and get in and stay in. Avirulence is when it can't attach. So that's what we talk about when we talk about getting rid of these viruses. The SARS-CoV-2 virus is an enveloped virus, and you remember what an envelope is on a virus. It's that outer piece of the host cell membrane. It can easily be gotten rid of by common disinfection. Clorox, Lysol, any of those things will get rid of that envelope, and then it becomes avirulent. It will not be able to attack us. So that is what you are doing for that.